Once again, welcome to Mr. Gos Biolectures. This Biolectures is very important. Make sure you subscribe, like, and share so that you can enjoy more of these Biolectures. Without wasting time, today's Biolecture is going to majorly focus on something we are calling the gene technology. We are going to look at now the gene technology. We want to look at the genes. Remember, a gene is just a section of the chromosome where the genetic material is contained. When I talk about the genetic material, I'm meaning what's called the DNA. You get? So you look at the technology. You want to look at if we apply some technology on the genes, is there anything that you can get that can be improved? In other words, hence called the gene technology. Now, remember this gene technology here, we're going to look at the major components here. We shall begin by defining what is the definition here. It's going to be the first point, center of gravity here. We shall have what to call definition. The definition what to call the what? Of the gene technology. Remember when we talk about gene technology here, we are majorly going to focus on something related to what we call keywords here. We shall see words like manipulation. That means manipulation. Manipulation. What are we manipulating? What are we trying to improve? We are improving something called the DNA. This is going to be called what? The DNA. So we are manipulating the DNA members. Then we are going to look at now when we manipulate, we can do this by altering. We have to alter. We must do what we call altering. What are we altering here? The DNA. You get? So during the altering, our changing of the DNA structure, we can either put in something or remove something. We can either insert genes. That means by inserting. That means by either inserting genes or by removing genes here. So why are we doing this one here? We are doing all that so as we can get an organism that is genetically modified. An organism with genetically modified structures. That means we are doing all these ones here to have what to call a genetically modified structure. Genetically modified structure here. A modified structure, organism, can also be called organism. We normally abbreviate as what we call a GMO, genetically modified organism. This organism here has what we call the special traits. How what we call the special traits. That means based on these keywords here, they can guide me to come up with a definition which is called the gene technology. You get? So gene technology refers to the manipulation of the DNA of an organism. You get? By altering it through inserting or removing DNA, I mean inserting or removing genes, so as to develop or to have or to come up with a genetically modified organism with special traits. You get? Within a given population. You get, members? So after looking at the definition here, we are going to focus on some other key areas. You have seen how to call the definition here. The next part, we need to look at something very important, which is going to be looking at the methods we normally use in genetic engineering. But remember, this gene technology here can also be called what to call the genetic what? Engineering. So we shall see what we call the methods. Methods used in genetic engineering. The method is not called genetic what? Genetic engineering here. Engineering, that is engineering. Or gene technology. So not get confused with members. We have what to call the word genetic engineering is the same as gene technology. You get? Because when I apply certain kind of technology onto the genes, is the same as someone who's engineering them. Engineering means that giving them that energy, that potential, that difference. You get? Now, when we finish this one here, of course, we look at these ones in details here. We shall again look at something very important, which is going to be now the application of genetic engineering. We see how is it applied in daily life. How do you apply it here? So it's supposed to be called the application of genetic engineering, application of the gene technology. Gene technology. That is very interesting, members. And then we start look at the clinical relevance. Number one is the clinical relevancy of this gene technology here. Or how do we apply it in drugs manufacturing? That means we use what we call the clinical relevancy. The clinical relevancy. Another is how do you apply it in the drugs? How do you apply it in the drugs? In making drugs, making medicine, that is making vaccines. You get? We shall look at all these areas. We see how you apply it here. Members, without wasting time, I want to begin by breaking one by one. And I'm going to begin by breaking by looking at something very interesting here. I'm going to begin by looking at something very interesting. What am I going to look at here? I'm going to begin with the number 2, which is called the methods used in genetic engineering. What do we do if we want to engineer the genes here? If we want to get an organism that is having the high quality genes, for example, want an organism having the genes that are for the skin color and want to obtain them, how do we do? Which procedures do you normally follow? Members, here is what we are going to follow, the several procedures. Hence, I'm going to begin by looking at that first one there. I'm going to begin with this one here, which is now going to be called now. Number 1 is done. I'm going to see what to call now the methods used in what? Genetic engineering. This is very interesting, members. Of course, members, if you are to do genetic engineering, there are some procedures we normally follow as individuals. We don't just come from nowhere, we follow some steps, members. Because if you don't follow these steps here, you may not come up with those genes that are genetically modified. The first thing we do something we normally call what we call the DNA isolation. You get? The DNA isolation. DNA isolation is something very interesting. I'm going to put it here. How about to call the DNA isolation? This is the first procedure we normally do. Just as human beings, the first thing that you have to do is that when you are trying to get one from the many, because the major focus here is looking at how we can remove the genes from a given chromosome. Sorry, from a given chromosome or from a given DNA. You get? Because in most cases, we don't only, okay, it depends on what you are targeting, but we normally remove a section of genes, not the whole chromosome, not all the genes, unless if you want to take all the genes from an individual. But in most cases, we normally focus on obtaining only specific genes from an organism. You get? So that means the first thing, if I want to remove a section of genes from a given chromosome, I first have to remove the chromosome from the what? From the nucleus. I remove the chromosome, so I remove the DNA from what we call the what? The nucleus. All those structures attaching onto it have to first remove them. Now, this is called the DNA isolation. You first remove it, isolate it from the other organisms. Then you have what called the gene cloning. You see what called the gene cloning. Gene cloning. Then we shall see what to call, after you have to call the gene cloning, what's going to happen now? We shall after, we have cloned this gene here, so now what to call the gene designing. The gene design. Then after designing what's going to happen now, we shall be able to do what we call now the transformation of the gene. Transformation of the gene. Transformation. Then last thing, what we're going to do here, of course, I'll just use it for breeding. Can I say breeding? Can I say plant or animal breeding? Can I say plant or animal breeding? So these are the major components of what to call genetic engineering. So when you do all these ones here, It means you are done with what we call what? Genetic engineering. Now, in today's bio lecture, we are going to majorly focus on what we call the DNA isolation. You get? We are going to focus on how can we isolate the DNA. This is going to be the major focus. You get, members? Now, let's begin. Now, I'm going to have what to call my DNA isolation here. It is going to be the major focus we're going to have here, which is called number 1. It is going to be the DNA what? Isolation. Something very interesting, members. It is going to be DNA isolation. Now, what comes into your mind, members, when you talk about isolating DNA? What comes into your mind, members? Now, just like the word isolation, that means we are trying to separate it from the what? From the other cellular complex. So as we can try to integrate it, or we can combine it with, if possible, the genes from another organism. So as we can now form what to call a modified, a combined gene. That's what we're going to look at it here. You're going to look at after DNA isolation here, gene cloning and recombinance. We're going to look at them at the same time. The DNA recombinant. In other words, the recombinant DNA technology. We're going to look at it here. Also, same as the DNA recombinant technology. Now, this DNA recombinant technology and also gene cloning, they look to be the same, but they are different. One of them comes the other. In DNA recombination here, technology, we are trying to get two cells that are come from different species. In other words, two genes that come from different what? Species. Two DNA. I get the DNA from bacteria. I get this is bacteria having a DNA. I also get one from the human cell. You as a human being, the mammal, any mammal. Then I mix them. When I integrate them, I mix or I combine them. You get? Of course, see, I'm going to get the genes of this of the human being of my interest. Let me say genes for the skin eye, for the skin color. So I'm going to integrate them. When I integrate them like this one here, what am I going to form, members? I'm going to form a product. This product I've formed here is called a combinant. You get? Technology. You get? It's what called a combinant gene or a combinant DNA. You get? Because genes are what called what? DNA. And what am I going to happen here? After getting it now, I'm going to multiply this one here. I'm going to put this into a host cell. You get? I'm going to put this in a host cell. So as it can multiply to form more other what? Cells, which I want. So the process of multiplying this gene recombinant here, or DNA recombinant here, in the host cell, is what we call what? The gene cloning. We shall look at it in details, members. Just giving you some recap. Now let's go to DNA isolation here. Now what are we doing here? This is just a mechanism, that a procedure or a technique used in the laboratories or in the industries. You get? To separate. You get? To separate or to isolate the DNA from other cellular what? Components. You get? We use it to do what? To separate DNA from other cellular components using what we call extraction buffers. Something very interesting here. We are trying to isolate. That's the key area. Let's look at the key areas here. We are trying to do what? We are trying to isolate. Trying to do what? The key areas in definition here. We're trying to do what? Isolate. This is what we call isolation. What are we isolating, members? We're isolating what we call the DNA. But what are we doing, members? We are doing this in several mechanisms. How do we separate it? By breaking, rupturing the cell membrane. The cell membranes must be broken down, must be ruptured. You get the point, members? That means we are doing isolating all these ones here using certain procedures. We're going to look at them here. You get? So, and now based on these, members, we're going to look at now how is DNA isolation. How is it? How does it take place majorly? Get members here. Get members here. Now we have some interesting here. This one is a human, it's a mammal, this is a human being. An interesting human being is here. This is the beautiful human being you are seeing here. This is the human being. This is his head, human being head. But on this head here, we are having some structures which are here. The ones you are seeing here, that's the hair. That is her beautiful hair. She has an interesting hair, members. But now I want to extract, when I fold up one hair, this hair here, what's moving, it is attaching here and then going to the head. But when I magnify it better, I will see that this hair here is attaching on a given cell. It is originated from the cell. Now, this is the hair cell we are seeing here, members. The hair cell. It has been zoomed. I've zoomed it so as I can see it clearly. This is the hair cell with its components inside here, which is called now the nucleus. Get the point, members? But now I want to examine further this hair cell here. What am I going to do here? I'm going to magnify it better. So this is what called now the next zooming. Have made it bigger. This is still called the what? The hair cell. Of course, once the hair cell, members, has components, what are we looking at? We are looking at how is DNA isolated, members. You get the point, members? We have begun the procedure now. You get? We have begun by looking at the hair. Of course, there are very many components where you can get the DNA. Or we can say samples through which we can have what called extracts. That's a brief note here. Samples. Of course, some can be gotten from the brain. We have the brain can give us a sample. We have what called the brain. The brain can also call the cells. The brain sample. We have what called the blood sample. We have what called the animal, or the plant tissues. All these can give out called what? The samples, the DNA. The saliva, or the saliva can give us members. You get? The lining of the buccal cavity can also give us. Get? All these can give us what we call the extract that we need to get the DNA. You get? Sweat even can give us any part of your body has DNA members. Let's continue here. We want to see how does the DNA, how do we isolate DNA from the cell? We are focused on what we call the what? The hair cell. Let's look at it, members. This is the hair cell we are seeing, members here. The hair cell has three major components here. First one is the DNA is located here. This is the nucleus, as you can see. And this one that you are seeing here is what we call the cell membrane. I think you can see that. The cell membrane. Let's see, members. Now, when you observe this clearly, members, I will see that inside here we have something very interesting. Inside the nucleus here, we have something very interesting here. This is called now the DNA. The DNA, of course, it's the cytoplasm here, the one which is inside here, the liquid inside DNA. And we want to remove this DNA here. We want to remove the DNA from here so as we can be able to integrate it or combine with the plasma DNA or the plasmid DNA or the DNA from another organism. You get? Now let's go on this. This DNA here can be isolated by three major processes. That means under isolation here, under DNA isolation here, we do it in several mechanisms. One, we have what to call, it can be done physically. You get? By crushing, you crush. When you crush, using a mortar and a pestle. You get? When I get my mortar here, I get my mortar. This is my mortar like this. I put my structure here. I begin crushing using what? The pestle. This is the pestle. I begin crushing it. I'll be able, this is what called the physical. That means when I apply the mechanical force here, it's going to be called the mechanical force now. I'm using the mechanical force. I'm using mechanical force. These are my fingers. This is the thumb. This one here. I'm trying to use the mechanical force of tearing up the cell structure. Hence, this is called what? The physical. You get the point, members? Meaning, how about called the physical? That's one. Then, secondly, we can use what to call the chemicals. Chemicals to break the cell wall. You get? This is going to be, for example, this is what we call chemical or chemically. For example, I can use what we call some detergents. Some reagents can be using some buffers. So this is going to be what we call the chemical. What's called the chemical. You get approach or chemically. You get? Then I can also remove this. I can also separate the DNA from here by using what we call enzymes. That means enzymatic. This is called what? Enzymatic mechanism. So this is what we call what? The enzymatic mechanism. Hope you're together, members. Enzymatic mechanism, what we're going to use there. That's what we're going to use there. That means we're going to see what's going to happen here, members. I want us to follow very clearly. We see what's going to happen during the process of isolating the DNA members. Don't forget we are still on what we call the gene technology. We are saying that gene technology is the process or the direct manipulation of the DNA of the cell of the organism. So by altering it through inserting or removing certain genes of the interest, so as to produce genetically modified structures or organisms with special traits. That's what we call definition. And so this gene technology here, it takes place in what we have what we call the methods you normally apply in gene technology here, which are here: DNA isolation, gene cloning. We can also add what we call the DNA recombination. We have what we call gene designing, the transformation, and what we call the plant or animal breeding. Then we're going to see what we call now application. But so far we are still on what? Method is using what we call genetic engineering. And we're looking at what we call the DNA isolation. It can be done in 3 ways. Physically, using the crossing or the manually, or using the manually, or we can say mechanically. Then chemically, using the chemicals like detergents, the chlorophenol chemicals. Then how about call it alcohol and so on, and also chloroform. Enzymatic, using 7 what? Enzymes. You get? And we are. Human being extracted one cell. This one cell here is what we are seeing here. We have magnified it. This is the one cell, contain the DNA, the nucleus, the cell membrane, and here are different ways we are trying to break what? Break something called what? The cell. You get? So after seeing everything here, we summarized everything. We've seen what we call the DNA isolation, which is a consisting measure of separate the DNA physically. That means by crushing it. Chemically, by applying some chemicals like detergents or buffers. Enzymatically, by applying some enzymes. You get? Like the ligase enzyme and so on, endonuclease enzymes and very many enzymes there. Now, we have seen how the enzymes, the physical mechanism and the chemical mechanism and the enzymatic mechanism have been participating in trying to act onto the what? Onto the tissue here. But I want us now to look at more so. We're going to focus on 2 mechanisms here. We're going to focus on what we call more so the chemical mechanism. Now, this isolation of the DNA depends on the type of the cell you're dealing with. Now, the cells you're dealing with here, they don't need a lot of enzymes to come and participate in, which I will just use a few of them and also more so the chemicals. Now, this is going to take me to this structure here. This is now the cell. It is going to undergo what we call some mechanical forces. That is physical, chemical, and then enzymatic. But I'm going to focus on 2 major focus here, components here. The enzymatic and also what we call the chemical mechanism. Now, in the chemical mechanism here, members, we are seeing what we call the cell lysis, the rupturing, the breakdown of the cell. And we're seeing in an individual here. What's the name of this individual here? This individual is someone who's very interesting. Who could be knowing this individual here? Someone help us. Now, this one is a chemical substance, and this one is going to be called a buffer. This is a buffer. This one is a buffer. You get? It's called a tritone. That's a trickstone. Trickstone X10. Or you can call it trickstone, or just say trickstone. This one is a detergent, not a buffer. Sorry. This is a detergent. This is a what? A detergent. So what's it going to do here? Members, this detergent is going to act on the cell membranes. The cell membrane, also what called the nuclear what? Membrane, by disintegrating it, by breaking it down. You get? By breaking it what? Down. Then also some other guy here, some other individual who's here sitting. What's the name of this guy here? You get? Some individual here is going to be called thylline. This is called the thylline, the amine tetraacetate. Now this one here is also going to break down. This one is a buffer. It is going to break down what to call the cell membrane. It's going to break and rupture it. It's going to remove this component of the cell membrane here. You get? They also have another component, members. It is called the sodium sulfate. Sulfates. This is what we normally call it SDS. SDS here. So that component, members, it is also very important. What does it do? It is going to participate in breaking the cell component here. The cell members want to call what? This one here. So most of these components here, detergent. So this one is like a buffer. This one also like a given buffer. They can deal with the breaking down the cell what? Membrane. So as they can be able to expose the components here. There is something I've realized here. This is the DNA. Of course, I want to this. Okay, DNA is okay. Let me take this to be the DNA. Okay, it's okay. It's okay. Now, I want to look at something very interesting here. We have finished what to call now the breakdown of these components inside the cell. What is going to happen here? That means we have a lot of impurities which are scattered within. The impurities are many there. That means if you look at the inside there, we have a lot of impurities. You get? Impurities are there. The proteins are there. We have this one here. We have what to call these are the sodium ions. The sodium, those are the ions. We have the proteins are here, the story proteins. Also have what to call the DNA. So we have very many components which are just scattered there, which are called impurities. What's going to happen here? We must first purify this what? Purify this DNA here by removing all unnecessary components. But before we do the purification, we must first allow these components here to separate themselves. I can say mechanically. How are we going to do? Before coming to what to call the purification here, we're going to first do something called the centrifugation here. So meaning after breaking the components here, we shall do what to call the centrifugation here. Centrifugation. What is centrifugation, members? This one is just the force, the application of the force, or what to call centrifugal force, that allows the materials to move at a high speed so as they can separate themselves. And those that are having a large molecular weight, they go at the bottom. Whereas those with a low molecular weight, they're going to go at the what? At the top. Now, after coming onto the top, now what is on the top is going to be called now the super tannant, something like that. What has come onto the top is what you're going to correct there. You get? So when you correct there, of course the DNA is also going to be having a low molecular weight. And therefore it's also going to be part of the what? Of the super tannant. You get? That means what you are having here what we are having here is called a super tanner, which is going to undergo what to call purification now, purifying now. You get? We want to begin removing those irresistible components. What are we going to do, members? How can we purify? Because our focus is want to remove the DNA only. We want to call this DNA here only. But what you are seeing here, this DNA here, members, when you look at this structure, it is having this what you are seeing here. These are called the histone proteins. Proteins. Then this coiling you are seeing what's going on, the real DNA. But now for us, we want to ensure that we remove what we call the proteins there. How do we do that? Of course, we're going to react to this histone protein, what we call the protease enzymes. We shall put what we call the protease enzymes. They are going to be combined there. I'm going to remove this by combining what we call the protease enzymes. Something very interesting. Now, what am I going to do here to some ions here? Of course, these are sodium and also some kind like, these are more so the sodium. This is what's called the sodium metals, the sodium element. That's called the sodium. And also, these metals here, what am I going to do? Most of those ions, what am I going to remove them? I'm just going to bring any buffer. For example, sodium or potassium acetate. You get? The potassium or the sodium. I have the ammonium acetate. I'm going to put that together there. I'm going to bring that kind of a buffer there. This buffer here, in a given solution, this buffer here, what's going to happen is that this buffer is going to break down what's called water to give what called ammonium ions plus what? Plus the ethanoate ions. Aqueous. What's going to happen is that this ammonium ion is going to work as a strong, in other words, as a weak base. That means it is going to receive all the protons, which is going to receive the protons from all the strong acids. So the strong acids which are here, in case there was an acid that was very strong, you get? It is going to be eliminated because we have very many acids here from the nucleic acid, from the RNA that was also broken down. So that means the nucleic acid, the hydrogen ions which have come from the acids are going to be buffered by combining with this plus hydrogen ions to form what to call what? Ammonium hydroxide. You get? Then this kind of the citrate, in other words, the ethanoic acid ions, the ethanoic ions here is going to act as a weak acid, and therefore what's going to happen here is going to receive what to call what? The hydroxide ions, form what to call ethanoic acid, something like this, or the sodium ethanoate, or something like that. Now, what's going to happen? That means that we have tried to buffer the ions inside the what? And the sum of the elements. That means the pH is going to remain normal for the functioning of other activities. And this, the moment we work on the pH, that means that the DNA is not going to face any problem. You get? The pH has been what? Stabilized. Hence, you shall not have any form of pH alternation, which is going to factor called what? The DNA. Now, after finishing this now, of course, we have tried to break some of the components. But what about some of the detergents? Remember, we have what to call this one here, detergents. Because remember, I have what to call detergents here. I'm going to put that to call what? Detergents. How can we remove detergents, members? I'm going to put it here, detergents. How can I remove detergents? I'm going to just use what to call certain kind of chemical substances, like the fluoroquine, the chloroform, or the chloropheno, chloroform phenol compounds. These are just go directly to remove what to call these detergents that we are using. Remember, when we are trying to break this cell here, so as we can remove the DNA, we apply the several detergents. Now it is time for us to remove these detergents using what we call the chlorophenol compounds. You get? Then, after removing these components now, we have already done what we call purification now. You get? Purification has been effected. What is going to happen next, members? We want now to do the last procedure, which is going to be now trying to remove, because the proteins have been removed. So we have the DNA remaining alone, plus what we call the what? Plus what we call the, of course, the cellular contents like the water inside there. What am I going to do here? I'm going to bring an ethanol, which is at a temperature of 020 degrees what? Celsius. And a sodium plus ethanol. And then I add onto this cell here, onto these components here. It is going to interfere. For example, this is the water molecule which is inside here. Hydrogen. This is water molecule inside there. I want to see how the ethanol is going to remove most of this water. So as we have a measurable amount of water, so the ethanol is going to majorly interfere with what we call the bonds here. You get? It's going to what we call the water bonds, hydrogen bonds here, and this is going to make the bonds to break. So when they break, they are going to be evaporated. It means the rate of movement or release of this bond is going to be faster. You get? And what to call this temperature here, and then we use what to call what? Ethanol. Sometimes we make what to call the chilled ethanol. The chilled ethanol. In opposed to what to call what? Presence of what to call the sodium ions. We call sodium as a metal. We shall be able to remove this, and therefore we shall now have what to call now a proper DNA. A proper DNA is going to be here. A proper DNA. You get? This is called a proper what? DNA we are having. So that means we have done now what we call the separation briefly. You get? Separation. You get? This is what we call now concentrating it. Here we are just concentrating it. You can say concentration of the what? Of the DNA. This is how to put the same as DNA concentration. So briefly, remember, this is what we call now the mechanism of isolating. So this DNA here, now it's going to go in the second part, which is going to be now the gene cloning or DNA recombination. You get? After getting it, we are going to look at the next biolecture, how we are going to break down. We shall apply some kind of specific enzymes which are going to break specific segments, having specific genes of this DNA. And then we also go and combine these with those segments from what to call what? From what to call another kind of organism of different species. So in the next lecture, we're going to look at now how we're going to break this DNA here, and after breaking it now, the segments we get, how are we going to combine them with those of other species to form what called a recombinant DNA, which is going to be inserted into what we call what? The host. And then it is going to be cloned, you get, for multiplication. And then we use it to form what we call other organisms or other species, or for industrial application, you get. So members, thank you very much for watching. That has been a brief lecture about what we call the gene technology.